patients today, so I decided to hold off.
Good morning. Welcome to another webinar from MPN Advocacy and Education International. My name is Ann Brazo. You'll see me sharing the screen with Dr. Aaron Gerds from the Cleveland Clinic, one of our pals and a frequent speaker with our group. Today, we're going to talk about clinical trials, and today it's specifically for patients and caregivers. And from what we understand, we have over 200 that have registered for this event. We will continue to do these online programs until further notice, but we think we'll just continue to do these forever, actually, because they have been so well received. You can sit at home and watch these or in your office, whatever you choose. We are looking at, we were going to have our first in-person event in Washington, D.C. on the 24th. That may change due to the numbers of COVID cases in this country. And of course, our first consideration is the safety and health of our patients. And so you will, if you've signed up or registered for that event, we may be making an announcement tomorrow to either confirm or to let you know that we have decided to do that online. So one of the reasons why we like to do these updates on clinical trials is to make sure that you are all aware of what our industry partners are up to, and you know, keep you apprised as to where their clinical trials are, phase 1, phase 2, phase 3. What's happening with their drug. There is some really good news, as we have told you repeatedly over the last several months, that we have several drugs that may be commercialized soon. And this is good news for the MPN community, since we only have 2 out there right now, and of course other therapies that are available. But the numbers of MPN patients is growing, unfortunately, but we want to make sure that everybody is cared for properly. The quality of care is great, and the access to treatment is available to you. So without further ado, I'd like to introduce Dr. Aaron Gerds again. Many of you know him. You've seen him at our events or have heard him at our online programs. And Aaron, can you share with us what's happening in your world at the Cleveland Clinic? And
Absolutely.
No further ado, here you are.
All right. Well, thank you very much. And thanks everyone for joining. It's a pleasure to speak with all of you today. It's fantastic. We have so many folks on the call. And again, looking forward to hearing your questions and spending some time with you. So what I'd like to do is just kind of cover a little bit about MPN clinical trials and kind of set the stage for what we'll talk about over the course of the webinar here. And, you know, really, again, we're focusing on the MPNs, of course, the pH negative MPNs, Philadelphia chromosome negative MPNs, include polycythemia vera, essential thrombocythemia, and myelofibrosis. And, you know, clearly these diseases have a lot of overlap, both clinically and genomically. And often we see folks' disease changing in their clinical form from, say, ET to myelofibrosis or even to accelerated phase disease. And that really tells us that there's some signals throughout this that really combine the two and common targets we can go after with our clinical trials. In addition to changing disease type, you know, there's a lot of overlap between the symptoms that people may experience or the clinical features that people may experience with these diseases, again, pointing to a common pathobiology. And this was first proposed back in 1951 by Dr. Demeshek, where he said, again, these diseases are so similar, there must be a common stimulus that connects them all. And then in 2004, 2005, 4 groups independently identified recurrent mutations in JAK2. And this was definitely a watershed moment because it led to the development of JAK inhibitors, kind of what we often use to treat patients with these days in terms of commercially available, FDA-approved type medications. And we know that in addition to JAK2 mutations, there are a lot of folks who have calreticulin and MPL mutations driving the JAK-STAT pathway within their disease. And since these all end up, no matter if it's a JAK2 mutation, a calreticulin mutation, or MPL mutation, it all ends up doing the same thing at the end of the day, driving JAK-STAT activation, it makes sense to target this pathway, independent of what the driver mutation is. And again, it's led to the development of JAK inhibitors where we use to treat these diseases. So currently we think about, for each of these diseases, the treatments that are available. And right here we have the treatments that are available for polycythemia vera down here at the bottom. So we use phlebotomies, aspirin, which is misspelled, hydroxyurea, ruxolitinib, and interferons, ruxolitinib being the JAK inhibitor. And then likewise for here, and we think about this, and the reason why we need clinical trials and we need new treatments is this is a workflow that we often refer to in the treatment of these diseases. This is called an algorithm. And this is an algorithm for high-risk polycythemia vera. So we start out here, these are patients who are over the age of 60 or have a history of a blood clot, and we think about what we're going to do for them. So right here, we think about, phlebotomy, and then adding a medication on either hydroxyurea or interferons. Then we monitor, and then if it works, great. If not, we go back to the list, and you may say that this list looks a lot like, after we determine intolerance, we think about, second-line treatments. Gee, that list of drugs looks a lot like the frontline treatment. So basically, we're kind of just like cycling through the medications in a big circle and not really innovating much, bringing new therapies here to patients that may need them that, the initial therapies don't work. So we always need new treatments. Likewise, treatment algorithms for, say, essential thrombocythemia tend to just to kind of recycle things. So we start, maybe with hydroxyurea or interferon. If it doesn't work, we switch to the other one, as shown in the right lower hand corner here. When we kind of cycle through the different medications we might use until we find that one that works. It's not really kind of moving forward or again, innovating the way we would like. In myelofibrosis, we often think about the symptoms that a person might be experiencing and then try to address all those individually. But more commonly, we think about disease risk in a big, massive algorithm like this. And the point I would like to make here is that if we look, so ruxolitinib or JAK inhibitors, JAK inhibitors, JAK inhibitors, JAK inhibitors. I mean, that's really kind of what we're seeing across the board here for the treatment of myelofibrosis. Just lots of JAK inhibitors. Again, nothing, we want to go beyond that. We want to do better than JAK inhibitors, although they've been revolutionary in the care of patients with MPNs, has moved the field forward dramatically, certainly a watershed moment, but we're always striving to do better. So we think about clinical trials, and this is kind of a snapshot of what of clinical trials we have available here at the Cleveland Clinic and ones we're thinking or looking into potentially opening. And this is the way we kind of organize these things. So a patient is diagnosed with an MPN, and the first thing you might see here are these green boxes. And so these are tissue collection and observational studies where we get samples from patients with MPNs and do research on them basically to better understand why these disease happens and different ways we can maybe be able to treat these things in new and innovative ways. So that takes a priority where we want to collect this information to improve things for patients. And then, for patients with essential thrombocythemia, we're thinking about we have a trial right now with a drug that is an LSD1 inhibitor. That's shown over here on the right-hand side. This trial with an LSD1 inhibitor, which is different from anagrelide, different than hydroxyurea, different than interferons, a completely different way of going after the disease. Even for MPNU, we have a clinical trial with a new and innovative medication that's different than anything else already available. We have forthcoming trials in polycythemia vera, all listed below here in the middle. Again, targeting hepcidin pathways or histone deacetylase pathways, things that are very different from hydroxyurea or interferons and stuff that's already available. And then for myelofibrosis, we're also looking to move things forward. JAK inhibitors often form the backbone of what we do in clinical trials, but we're adding in new medications, again, to try to get these JAK inhibitors to work better or get deeper responses or regain responses that have been lost. And when we think about trials in myelofibrosis, there's really kind of a couple of different bins we want to think about in terms of the clinical trial space. So there are patients who have never been treated with a JAK inhibitor. And then there's patients who've been treated with a prior JAK inhibitor. And that's really the kind of the main difference there when we're looking at the different clinical trials. We often think about, what a person's platelet counts might be, because that could determine what trial they're eligible for. So for example, patients with platelet counts less than 50,000, pacritinib is a clinical trial, the Pacifica trial, enrolling patients right now. For patients with a very high platelet count or normal platelet counts, we think about ruxolitinib plus CPI 0610, which is, and so we kind of parse out different treatments in that manner based on platelet counts. Another way to kind of parse out the different clinical trials is whether patients are anemic or not, and is that their major issue, and can we address that directly with new medications like luspatercept. And then, certainly after progression, where the disease has moved past JAK inhibitors, and again, trying to hit new pathways that are independent of JAK inhibition. So really that's kind of the main focus here of a lot of these clinical trials. So again, that was just kind of to get everyone's set and think about, well, where is the current treatment of myelofibrosis, PV, and ET? We have hydroxyurea, we have interferons, we have anagrelide, we have a few other treatments that are available in addition to JAK inhibitors, but really that kind of forms the bulk of where we are now. And so by recognizing where we are now, we can look forward to where we need to go in the future. And a lot of that is targeting pathways that are different than JAK-STAT, combining JAK-STAT inhibition with these new targets, and to really kind of deepen responses and certainly move the field forward. So that's really what I have prepared for kind of the introduction portion of this. And I will stop sharing these slides. And, I know Ann has a selection of folks that are all prompted up, ready to go to talk about a lot of these new and innovative strategies.
And before I introduce the first person, for the first industry partner, Aaron, a lot of our patients are just so excited and so anxious for new drug therapies. But are you finding, and I believe this to be true, but you might be able to elaborate a little bit on this, that you're starting to use more cocktails, this to address the anemia, this to address,
yeah.
Yeah.
It's really moving towards individualized care. So, we'll take a look at a patient and say, well, yeah, your most pressing issue is anemia right now. And so let's try to develop a therapy that specifically addresses your anemia. Well, the next patient might have their most pressing issue may be a return of their splenomegaly after starting a JAK inhibitor. So we say, well, gee, you are still benefiting from the JAK inhibitor. Perhaps we should just add something on top or an add back strategy where we can then regain the response. There's another trial where we're actually starting to incorporate mutational profiles into the treatment algorithm. So the example is the MPNRC study targeting a disease that has an IDH2 mutation with an IDH2 inhibitor. So we use ruxolitinib plus the IDH2 inhibitor specifically for those patients whose disease contains that mutation. And really, you can see we're on the precipice of this kind of precision medicine or tailored therapy era, where we're going to be looking at an individual and their symptomatology, as well as the mutational profile within their disease to determine the best course of action going forward. So these are kind of the first forays into that, really. And then primarily, kind of the bulk being these combination therapies.
Yeah, I think so too. I mean, obviously. And so if you'll stay around while we continue this, because I know we're starting to get some more questions from the registrants, the patients and caregivers. But thank you very much. And for that summary, that was great. And we'll move on to hear from Imago Biosciences from Dr. Wanchen Hong, who's an MD and Chief Medical Officer at Imago. Hello and good morning. How are you?
Hi, Ann. Nice to see you.
Good to see you too.
Let me share my screen.
It's there.
What is that?
You can see it. Yeah. Well, yes, there it is.
Is it in the right format?
Yes.
Okay, great. So thank you, Anne, for the opportunity to present some updates from Imago Biosciences, and our lead candidate is called Bomedemstat, which Dr. Gerds had mentioned earlier in his talk, and it's a LSD1 inhibitor. So quickly, this is a quick snapshot of our pipeline. As you can see, essential thrombocythemia is the lead indication, and currently we have a phase 2 study that's enrolling, and we hope to share additional updates later this year at ASH. And I'll also be showing a little bit some of the highlights from previous presentations that were from earlier this year. We're also evaluating Bomedemstat in myelofibrosis, where we also have a phase 2 study. The enrollment for this study is complete, and again, we hope to share additional updates later at ASH this year. We also have a couple IST programs that we hope to initiate, the Bomedemstat plus ruxolitinib combination. Also, the study has not started yet, but hope to initiate that maybe later this year, as well as a polycythemia study that's currently enrolling. The last two on this slide are new chemical entities, so these are still brand new, not in the clinic yet. So first, I'm going to just go through this rationale for LSD1 inhibition in MPNs. So LSD1 regulates the proliferation of blood stem cells and is also essential for their differentiation into megakaryocytes and granulocytes. And so if you inhibit LSD1, this has been shown to reduce the symptoms of MPNs, as well as lower the number of cells and mutations that drive the diseases. And that's shown below in the figure, where you have a hematopoietic stem cell on the left side, and then it becomes, you need LSD1 to have activated megakaryocytes, but when you inhibit LSD1, then you reduce growth factors as well as your reticulin and collagen, and that would reduce bone marrow fibrosis. And this is important for MPNs, especially myelofibrosis, so reducing splenomegaly and extramedullary hematopoiesis. It also, if you inhibit LSD1, this decreases the inflammatory cytokines. And this is also important for a lot of the symptoms that patients have, and that includes fatigue, anemia, pain, itching, fever, as well as night sweats. One other property of the LSD1 inhibition is really trying to reduce the malignant cell population, and this hopefully can actually change the disease, the natural history of the disease. And this is we're still hoping to show this through our trials. So Bomedemstat is a novel LSD1 inhibitor for the treatment of MPNs. It's otherwise known as IMG 7289, and it's an irreversible inhibitor that was discovered by Imago. It addresses the limitations of other LSD1 inhibitors, such as it minimizes crossing of the blood-brain barrier. And in different models of MPNs, and one is shown on the right side, LSD1 inhibition reduces some of the key hallmarks of the disease. And you can see in this mouse model of JAK2 V617F mutated mice, the mice that were treated with Bomedemstat survived longer, where the ones that did not receive treatment did succumb to the disease. And so far, we've demonstrated activity, and the drug is well tolerated in greater than 150 patients. And then we've been using platelet count that serves as a biomarker of Bomedemstat activity in megakaryocytes. And this allows for personalized dosing so that the drug is titrated according to the patient's platelet count. We've also have some regulatory designations from the FDA and EMA, including the orphan drug designations and fast track in the US for ET and MF, orphan and prime for MF in the EMA, as well as orphan drug designation for ET in the EMA. So first, I'm just going to quickly walk through some of the data in the myelofibrosis study. And this is a study, phase 2 study in advanced myelofibrosis. This is a global study with 89 patients enrolled so far, and actually the enrollment is now complete. Patients are dosed with Bomedemstat orally. This is once daily dosing, and patients who are eligible require a platelet count of greater than 100,000 and are resistant to an approved therapy. The objectives of the study include safety and tolerability, symptom reduction, spleen volume reduction, and there's also additional secondary and exploratory endpoints. The demographics, the key demographics in the study are 57% are high risk by IPSS, 67% have 2 or more mutations that are associated with MF, and then 44% have mutations that are high risk for AML transformation. This is showing the data from that was presented at EHA this year, and this is the total symptom score at 24 weeks. So all patients that had a TSS greater than 20 at baseline, this is showing the subgroup of patients that had the higher TSS scores. So you can see that the majority of patients, 15 out of 16, had a decrease in TSS at 24 weeks. And about 31% had a decrease of greater than 50% at 24 weeks. And that's shown in the figure on the right side. In terms of spleen volume reduction, this is again, a subgroup of patients that we have the data for. And the majority of patients, or 89%, had a decrease in spleen volume at 24 weeks. And again, this is shown on the right side. And then if we look at, the threshold of 35%, fewer had met that threshold, but at least most of the patients did have a decrease in spleen volume. For the Bomedemstat and ET study, again, this is a phase 2 study in high-risk patients. Again, this is a global study, and we currently are estimating to enroll 60 patients, and enrollment is currently ongoing. Patients are dosed with Bomedemstat by mouth once daily, and patients who are eligible for the study are high-risk patients who are intolerant or resistant of standard of care, which is most commonly hydroxyurea. The objectives for this study include safety and tolerability, reduction of platelet count to less than or equal to 400,000 in the absence of thromboembolic or hemorrhagic events and progression of disease. And we're also looking at the reduction in mutant allele frequency in the study. So the primary objective is to reduce the platelet count in the disease where the platelet counts are very high. And at the time of the EHA presentation, 83% of the patients who were dosed for at least 6 weeks achieved a platelet count of less than 400. And the mean reduction of platelet count was about 550,000 at week 12. You can see in this figure, the yellow dotted line represents the 400 threshold. And you can see, many patients start high, and then, when they're off their prior therapy, the platelet count can go up. And then once they start the therapy, which is here in this line, the platelet count starts to go down. It does take some time for the platelet count to go down, but you can see that the majority of patients do eventually achieve platelet counts of less than 400,000. And what's also important is that that Bomedemstat is able to lower the white blood count and also maintain hemoglobin levels. So here, again, looking at a similar graph with white blood count plotted. Some of the patients have elevated white blood count. Here we show that at level of 10. And when they're in the screening period, the white count can go up because they're not on any drugs. And then when they start Bomedemstat, the white count can come down. So for the patients who had elevated white counts at baseline, 80% or 4 of the 5 patients returned to a threshold of less than 10. And also important to note that we did not see any neutropenia in this study. Also for hemoglobin, again, this is important to see if the Bomedemstat induces anemia. You can see that the hemoglobin is fairly stable, and so it maintains hemoglobin levels. In terms of the total symptom score, we see changes in total symptom score at 12 weeks, and this is in the subgroup of patients who had baseline scores of greater than 10 at baseline. So we have few patients here, but you can see that the majority of patients had a decrease in the total symptom score, and 2/3 of the patients had decreased more than 10 points. And so that shows that the Bomedemstat can reduce the symptoms in patients with ET. So in summary, Bomedemstat or IMG 7289 is an irreversible inhibitor of LSD1 that's being evaluated in MPNs. Overall, Bomedemstat is generally safe and well tolerated in greater than 150 patients to date. Bomedemstat showed improvement in symptom scores, spleen volume, and anemia. I didn't get to have time to show the data today, but we show that there are decreases in mutant allele frequencies in 44% of the patients. This is from the EHA poster. And there were no new mutations or transformation to AML in the study. In the ET study, Bomedemstat monotherapy has shown improvement in platelet and white blood cell counts while maintaining stable hemoglobin. And also Bomedemstat demonstrated improvement in symptoms. So in terms of development plans, MF study has already fully enrolled, and we are hoping to initiate the combination of Bomedemstat and ruxolitinib as an IST study soon. And the ET study remains open for enrollment, and because based on the promising data we've seen so far, planning for a pivotal study is underway. I just wanted to thank all the study participants and families for supporting our program. Thank you.
Thank you very, very much. That was so very informative, and we're very happy to hear the progress of the drug because, as I said earlier, we're in dire need of new therapies for MPN patients. We do have a few questions, and Dr. Gerds, if you're there, yeah, there you are. Is there evidence that would show if a patient has been initially diagnosed, let's just say with MF or with one of the MPNs, and they're on a medication that is reducing and actually eliminating some of the symptoms, let's say, for example, hydria, would a physician take a patient off of the drug?
Yeah, there's certainly a couple of instances we can think about where a medication might change the disease. Yeah, I think the one that kind of caught a lot of people's interest, at least at the most recent American Society of Hematology annual meeting, was interferon and the discontinuation study that was done is still ongoing in France, where people got into such a remission where they didn't need, they went on a drug holiday and for extended a period of time. So I think that's one situation where a drug induces a really deep remission. We've seen that with some of the other clinical trial medications. I have a patient that we had on the myelofibrosis trial for the LSD1 inhibitor that was just discussed, whose counts went in really, really deep remission and actually went off treatment for a while, didn't need treatment for like a year plus. So I think it definitely can happen. The other kind of reason why you may need to adjust as the disease is changing, it's type, right? So if a patient is on hydroxyurea for a very long time for their polycythemia vera, and their polycythemia vera transitions to myelofibrosis, post-PV myelofibrosis, you may not need hydroxyurea to control the counts for a period of time, and that might be your first clue that the disease is changing.
Are there any clinical trials that allow children with MPN?
Yeah. That's a tricky one. There aren't a lot of trials that, MPNs in children aren't terribly common. They're not common in adults, right? So they're even less common in children. So I think that makes it hard to do clinical trials because you're just not going to get the numbers of patients. Most clinical trials start at the age of 18 and work up from there. So it does cover the AYA or adolescent and young adult population, which I think is excellent. And then a lot of times in the everyday care of patients with MPN who are under the age of 18, we're often using adult therapies in that space just because there are limited trials in that population.
Thank you. Okay, so we'll move on, and thanks again, Chen, that was great. We will move on now to KARTOS Therapeutics, who is one of our partners, and to Nish Saini. And he is the VP of European Medical Affairs. And Nish, thank you for joining us. So I'm glad to see you here.
Thank you, Anne, for the opportunity. I'm going to share my screen now. If you could just confirm that you can see the screen in full display mode, that would be...
There it is, yeah.
Right. Okay. I'm just going to go into presentation mode. So again, can you see the screen still?
Yeah.
Perfect. All right. Well, good morning, good afternoon, everyone. My name is Nish Saini, so I work for KARTOS Therapeutics in the medical affairs team, and really thankful for this opportunity today to speak to you and provide this brief study update on the clinical trials that KARTOS is running in myelofibrosis. So I imagine many of you may not have heard of KARTOS before. We're a relatively small privately owned biotech company based in the San Francisco area, though I actually work out of London in the UK. And our company's focus really is the development of novel targeted therapies that have the potential to meaningfully improve the lives of patients with cancer. We're actually currently developing an oral investigational agent called Navtemadlin, previously called KRT-232. And we're actually exploring Navtemadlin across a number of blood cancers and solid tumors. But a major focus of our development of the compound really is in myelofibrosis, where we have currently 4 ongoing clinical studies. Now, Navtemadlin targets a protein called MDM2, and MDM2 is a key regulator of another very important tumor suppressor protein that you may have heard of called P53. And on this slide, you can see a bit more detail about P53. P53 is often referred to as the guardian of the genome, and it plays a really important role in the natural life cycle of all cells within the human body. Now, when P53 is activated, for example, in response to DNA damage or oncogene activation that may occur in a cancer cell, P53 in turn activates a number of genes that function to block cell division and induce cell death. Now, it's this ability of P53 to block cell division and induce cancer cell death that actually relies on quite a delicate balance between its own levels and the levels of another protein, MDM2. And MDM2 is important because it can block P53 activity through a number of different mechanisms, which I won't describe in detail today. But in other words, MDM2 actually puts the brakes on P53's ability to drive cell death. Now, unfortunately, it's been shown that in myelofibrosis, cells can actually abnormally increase their MDM2 levels and shut down the ability of P53 to fight cancer. So KARTOS is exploring the potential of our investigational agent, Navtemadlin, to inhibit these high levels of MDM2 that are found in myelofibrosis, and hopefully thereby restore the ability of P53 to kill cancer cells. So we actually presented some quite encouraging initial data from a clinical study of Navtemadlin in myelofibrosis at the EHA Congress in 2020. And as a result of that data, we've actually opened now a large randomized global phase 3 study called the BOREAS study that will compare the safety and efficacy of Navtemadlin with best available treatment in patients with myelofibrosis whose disease has progressed or failed to respond to a JAK inhibitor. And I'll provide a little bit more detail about this study in the next few slides, but I also wanted to highlight 3 other studies that we have ongoing exploring Navtemadlin in different settings. This next study, we call it the 113 study, and you can see the NCT numbers here if you need more information. But this study is looking at combining Navtemadlin with another investigational agent belonging to a class of drugs called tyrosine kinase inhibitors. And we're exploring this combination in patients who are relapsed and refractory or intolerant to a JAK inhibitor. Our third study is looking at the combination of Navtemadlin in ruxolitinib in patients who have a suboptimal response to ruxolitinib treatment alone. And we even have another study, a 4th study, we call this a window of opportunity study, which is actually looking at Navtemadlin in a frontline setting in patients who have yet to receive a JAK inhibitor. Now, all of these studies are open at multiple sites in both the US and in other countries across the globe. And I've got a bit more detail in an upcoming slide about the BOREAS study, but you can obviously find more details about all these trials on clinicaltrials.gov using the NCT numbers that you can see here on this slide. But I just wanted to give you a little bit more background and information about the BOREAS study, our global phase 3 study of Navtemadlin in patients with JAK inhibitor relapsed refractory myelofibrosis. So BOREAS is the name we've given this study, and it's an acronym that comes from the term blocking MDM2 to reactivate P53 in myelofibrosis. So it's in very small text here in the upper left-hand corner of the slide. Now, BOREAS is actually the Greek god of the north wind, and we gave the study this name because it's quite consistent with the exploration and navigation theme from which our company's name, KARTOS, is actually derived. And what you see here in the imagery are the links between BOREAS and Aurora Borealis, the northern lights. So this is a very high-level summary of the study design. So this is a phase 3 study. We plan to enroll 282 patients with myelofibrosis whose disease has progressed or failed to respond to a JAK inhibitor. And we're enrolling patients in 180 sites across US, Europe, and rest of the world. And I've got a site map for you coming up, which will give you more detail, but we're enrolling this study in 25 different countries. Now, patients are going to be randomized in a ratio of 2:1 to either treatment with Navtemadlin or physician's choice of best available treatment. And after 24 weeks of treatment, we're going to evaluate the efficacy of Navtemadlin by comparing the reduction in spleen volume and the improvement in patient symptoms across the two different arms that you see here. So this slide gives you a little bit more detail about all the different countries where the study is enrolling at the moment. There are 14 sites that are planned to be open in the US, and 12 of those are currently open as we speak. And of course, you can find more details, as I said, on clinicaltrials.gov by searching for this NCT number. But I also wanted to highlight that we have actually launched a website for the study called boreastrial.com, and that also will provide you with a lot more background and information on the study. So that was a very quick overview of KARTOS Therapeutics and our development plans in myelofibrosis. We're a very small company, but I think you get a sense that we have a lot going on, and we have a very strong commitment to the development of novel treatment approaches in MF. So thank you again for the opportunity to speak today. We're really excited to engage with you, the patient community, and obviously to collaborate with groups like MPN Education and Advocacy International. And we look forward to continued partnership moving forward. Finally, if you want more information about KARTOS or the study I mentioned, we've got a couple of websites that you can go to. boreastrial.com, which is currently accessible if you live in the US. And we have kartosthera.com as well, which will give you more information about the company. And if you choose, you can also follow us on Twitter using the hashtag KARTOSthera. So thanks very much for the opportunity again, and I'll hand back to Anne.
Thank you so very much, Nish. This is excellent information, and to all the patients that are watching and listening right now, I mean, this is pretty profound. 17 years ago, when I got into this space, there was nothing, very little to nothing happening. And I know Dr. Gerds is agreeing with me because there was just so little happening. This is remarkable as far as I'm concerned, Dr. Gerds.
Yeah, I mean, we're thinking about attacking these diseases in a lot of different ways, and I think MDM2 inhibitors are an interesting way to go about things, because we're inhibiting the inhibitor of the thing that can kill the cells, which is, you have to think, it's almost like a double negative, but it clearly, this class of medications clearly can kill off disease, which is something that a lot of the therapies we have available outside of bone marrow transplant can't do. And I think that that's the really exciting part. Can we reverse the disease course, or turn back the clock, if you will, for someone's disease, as opposed to just managing it? We really want to push it back. And I think that's the real thrust of a lot of these therapies that we're developing right now in partnership with our commercial partners and basic scientists and otherwise. So I think that's really the exciting piece.
Yeah. So a lot of patients are asking, what is it going to cost them to be in a clinical trial?
Yeah, I think that's a great question about the mechanics of a clinical trial. So the simple way of breaking it down is everything that we would normally do to care for your MPN is covered by insurance. It's submitted to your, doctor's visits, basic labs like blood counts and such, all billed as standard of care to your insurance. And then anything that's additional is billed towards the clinical trial. Extra blood draws, extra doctor or nurse visits, certainly the medication itself, extra CAT scans or MRIs that are being done. So all that's billed to the trial. So from a perspective of your insurance, it should not look any different than normal standard care. Also, there are sometimes incentives, additional perks that are part of a clinical trial. For example, here we like to offer valet parking for free to all of our clinical trial patients because we know they come much more often. A lot of trials that require you to be here for a longer period of time may offer, allowances for food and even hotel stays to kind of cover those costs, those additional costs. So there are additional resources to help ease the burden of clinical trials. Lastly, there are some institutions that actually have foundation medicine to help cover, foundational, not medicine, but foundational funds to help cover additional costs that incur via clinical trials and to encourage trial participation. Oklahoma is one that I'm aware of that is a really nice a piece to what they're doing with their clinical trials.
Yeah, we do discuss this at our events quite often with patients that they're, and I discuss it with our partners, our industry partners, that, we will certainly, get the word out about your trial. And I think there are a lot of people that would like to participate, but have, some challenges in doing so, such as daycare, letting their jobs know that, they have to leave on that certain day, and don't want their employers to know that they're sick or that they have something wrong. There are lots of things, but what I love about our industry partners right now is they're extremely open to this conversation of what else can we do to help. And I've had numerous conversations this year with each group, actually, discussing this very topic of how do we first of all, how do we get a broader section of individuals into clinical trials, and of course through education and communication with their physicians and with us and other organizations, but also how do we negate some of the disparities they may have in getting to the trial, and how do we manage that. And so those are big issues on our table right now that we hope to find some solutions. All right, another question, and then we'll go on to our next partner. And this was discussed at a meeting I went to several years ago, and a gentleman presented, and it was very interesting. And he was actually trying to get a movement together to take away the placebo part of clinical trials. And I get it. I totally get it, because I would probably sign up for a clinical trial, but I would really want the drug instead of placebo. Yeah. Can you explain that a little bit?
Yeah. So, the reason for the placebo is to try to eliminate all bias. The example I always give is we did a trial a long time ago with a drug that is no longer in development, but on the physical exam, the docs would measure the spleen. And if you look at the chart of the spleen sizes, they all shrank universally in this very small trial. But then we also got imaging, but the docs were blinded to the imaging. They didn't see the CAT scans or MRIs. In the CAT scan and the MRIs, all the spleens got bigger. So clearly we're biased, even if we don't, we want to think the best of things. And so it's to really eliminate that bias so we can get a true sense of the efficacy and safety and side effect profile of a medication. Because a placebo should not cause anything. It should just be a placebo, right? Outside the placebo effect, of course. Now, I think a lot of but it is hard. Like, you want to get a therapy. And, I think if the normal standard of care is just observation, then placebo makes sense to a certain degree, right? Because if outside the clinical trial, you just be on observation and you're getting a sugar pill, you're getting the appropriate treatment at a minimum. I think what happens now is we're actually randomizing a lot of patients to best available therapy. So the idea is, well, we got this list of commercially available agents and things that we can do, but there's this new medication. So we're going to randomize you, meaning half the people randomly get the new treatment, but half will be randomized to whatever you were going to do anyways outside the clinical trial. And I think that's more appealing. At least you're getting something. You're going to get what would normally be given to you. A lot of these, combination studies now, especially upfront ones, are using placebos in a slightly different way. So say your normal treatment would have been ruxolitinib. So on a trial, you may get ruxolitinib plus placebo, which you were going to get anyways outside the clinical trial, or ruxolitinib plus the new drug. So again, and a lot of these trials will allow often for crossover at some point. So if you are on the placebo arm, you can get access to this new medication at some point in time. So I think that's another way of making sure that everyone is adequately treated and you're getting something. Worst case scenario, you're getting what you would normally outside the clinical trial. And I think that's the way to look at it. And the placebo gives us really the power to truly understand the efficacy and safety and side effect profile of these medications without minimizing to the best of our abilities any personal bias we might have.
Good answer. That was very good. Okay, so before I move on to our next partner, I just want to remind all of you that you can post your questions at any time and we will get those and be able to ask Dr. Gerds or one of our partners. And then also any links that are mentioned here today, we will post those and follow up with, you can follow up through us that were mentioned today. You can let us know, or we will let you know, I'm sorry. Okay, so hang in there, Aaron, and we will be back. So our next industry partner is from AbbVie, and Brian Murray has been with us before. He's a pharmacologist and MBA. And Brian, if you would like to share your screen, and we will be anxious to hear from you.
Thanks so much, Anne. Can you hear me?
Yep.
And can you see my screen?
Can do that, yes.
Okay, great. Let me just make these, let me just blow this up a little so you can see the slides a little better. Okay, there we go.
Perfect.
All right. Well, thanks so much, everybody, for being here. Thanks so much to Anne, Kathleen, MPN Advocacy and Education International for yet again giving me another platform to discuss some of the things at AbbVie that we're working on in the myelofibrosis space. Today, I'm going to be talking to you all about a novel agent called navitoclax in combination with ruxolitinib in patients with myelofibrosis, in particular 2 phase 3 studies that are currently ongoing and looking for patients. And so without further ado, get right into it. Just a moment. So briefly about AbbVie, many of you have heard of AbbVie. We have a very strong presence in hematological malignancies. We have many therapies under investigation. We have approved therapies as well. Some of you have probably heard of venetoclax in CLL and AML. Navitoclax is a very close cousin. And so, we hope to continue advancing these therapies through the clinic to give more options to patients who are in need of them. Before I discuss the studies, just a few disclaimers to mention. The navitoclax, the compound that I'll be discussing, is not FDA approved. It's in any therapeutic area. It is currently under evaluation for safety and efficacy, and is purely investigational at this point. All right, so before I discuss the phase 3 studies, I want to talk a little bit about the mechanism of action of navitoclax and how it works, and why we're studying it in combination with ruxolitinib. So navitoclax affects the cell death pathway involved with that cancer cells use to keep themselves alive. So cancer cells will hijack certain proteins within the cell death pathway to keep themselves living and evade the normal mechanisms of death that generally occur normally in the absence of any overactivation. You saw Dr. Gerds earlier present a diagram on the JAK-STAT pathway, and as you're all familiar with, overactive JAK-STAT signaling is what drives myelofibrosis. What you might not know is that JAK-STAT pathway overactivation also leads to the production of a pro-survival protein called BCL-XL. Navitoclax inhibits BCL-XL. And what happens is when you inhibit JAK2 with a JAK inhibitor such as ruxolitinib, what it does is it shunts that pathway and it leads to decreased production of that pro-survival protein. Now, what does that mean? What it means is the cell is increasingly vulnerable to a BCL-XL inhibitor, such as navitoclax, because the cell death process is very carefully regulated. It's a very delicate balance. So if you have a preponderance of the pro-death proteins, it tips the seesaw in favor of cell death. So when you inhibit this protein with navitoclax, what happens is that the pro-death proteins are able to do their job and cause cell death. And this applies to cells in myelofibrosis. So we have preclinical models, the data which that I'm not able to share to you today, that show a synergy between navitoclax and ruxolitinib in these early phase models. And on the basis of this and other models, we initiated a phase 2 study, which I won't be discussing today. And we've generated some data from that study recently presented at EHA and previously at ASCO. But the studies I want to discuss with you today are about the phase 3 studies, which are active and ongoing. We haven't presented any data from these studies quite yet, but we are actively seeking patients. And this is meant to be informational for you to see if you may be interested and potentially eligible for these studies. So the first study I'll discuss is a phase 3 study combining navitoclax and ruxolitinib in patients with myelofibrosis, and we call it Transform 1. Here's a simplified study design. This is a phase 3 randomized, double-blinded, placebo-controlled study. So, both the participants and the investigators are blinded to the treatments, we have a placebo-control group which Dr. Gerds discussed just a couple minutes ago. So we're looking for 230 patients. We'll randomize them into one of 2 treatment arms on a 1-to-1 fashion. So you'd have an equal chance of receiving either the experimental therapy or the placebo therapy with ruxolitinib. The experimental arm is navitoclax, which is an oral agent given once daily, combined with ruxolitinib twice daily. And the placebo arm involves the placebo, of course, along with ruxolitinib as well. And this is continued until disease progression. And will be follow-up as well. Here's some of the eligibility criteria. I know it's quite a bit, but I wanted to present some of these just to give you a sense of the sorts of patients that would be eligible for this study. So I'm not going to read through all these, but I'll hit on some of the highlights. So as I'd mentioned, patients with primary or secondary myelofibrosis are eligible. You have to have intermediate 2 or high risk status as defined by the Dynamic International Prognostic Scoring System Plus, or the DIPS Plus scoring system, and as well as meet some qualifications related to the symptom assessment via the MF symptom assessment form. You have to also present with splenomegaly, and which is defined there. I won't read through that. And no active malignancy other than MF in the 2 years prior to screening. There are some exceptions to that in the footnote there that I won't read through. Patients cannot, going to the right side of this slide, patients cannot be candidates for stem cell transplantation at study entry or have had prior treatment with JAK2 inhibitors. Remember, this is a study in patients that have not been exposed to a JAK2 inhibitor, such as ruxolitinib. No prior treatment with BH3 mimetic, which is another term for a compound similar to navitoclax or BET inhibitor, and patients cannot have had leukemic transformation. I won't read through the others, but I'll refer you to a website where you can get more information, including the information that's been presented here. Before we discuss that, though, I want to talk to you about the second phase 3 study that I mentioned, which we call Transform 2. And this is a study evaluating the same combination that I mentioned, navitoclax and ruxolitinib, compared to best available therapy in patients with relapsed or refractory myelofibrosis. So this study is different from Transform 1 in that these patients have been exposed to ruxolitinib and have progressed. And in this case, we're looking for 330 patients randomized 1-to-1 to the experimental arm, which is the exact same as the Transform 1 experimental arm, navitoclax and ruxolitinib, and a control group, which I'd mentioned was best available therapy. Now, best available therapy in this case is up to the investigator's discretion. So you could receive ruxolitinib or continue the ruxolitinib, regression, hydroxyurea, interferon, danazol, or fedratinib, which many of you are familiar with. And again, this is continued to the end of treatment or disease progression. I've shown the eligibility criteria here, very similar to Transform 1 with a few exceptions, and I'll call some of those out. So like Transform 1, we're looking for intermediate 2 or high risk myelofibrosis patients. and patients who have developed splenomegaly. And then the patients have to have had, as I'd mentioned, prior treatment with one JAK2 inhibitor that was either stopped due to refractory disease, relapse disease, and then the various time constraints and parameters that are listed there. Similar to Transform 1, you can't be a candidate for stem cell transplant. And also similar to Transform 1, patients have to have platelets above 100,000. I didn't mention that previously, but you have to have platelets over 100,000. Dr. Gerds mentioned some of the various trials that he has open with the various platelet cutoffs. And then the rest of the exclusion criteria on the right-hand side of the slide are very similar to Transform 1. Here's some of the endpoints or the measures of efficacy and safety that we'll be evaluating in the trial. Many, if not almost all of you, recognize these as quite standard for myelofibrosis trials. The primary endpoint in both of these trials is spleen volume reduction of at least 35% week 24. And then for secondary endpoints, we're looking at total symptom score, various measures of survival, and then we're also looking at quality of life measures as well, including fatigue, physical functioning, and others. And then, of course, we'll be evaluating the safety of each of the agents, including the treatment emergent adverse events and the various other measures there. So lastly, before I end, I want to mention a resource, which I think you might find very handy. There's a website that you can, that's publicly accessible. It's called myelofibrosis.research.com. And on this website, you can learn more about these trials that I discussed. It has more detail, including some of the details I just covered. It also has a questionnaire to help you determine if you might be eligible for any of these studies. Very handy, just it's a series of questions that you answer yes, no, or various other answers, and then it gives you a sense of if you might be eligible if you're interested in these studies. There's also a handy Google map that's embedded into the website that has our current study locations throughout the country. We have sites in over 20 states. We're currently, we're continuing to activate sites throughout the country as well as globally. And then there's a frequently asked questions section to address any potential questions that you might have and contact information if you want to get in touch. And so I think I will stop there and I will turn things back over to Anne. Thanks, Anne, for the opportunity, and I appreciate the chance to speak about our program.
Thank you very much, Brian. That was excellent. Wow, I'm overwhelmed, to tell you the truth, because there's just so much going on and going in the right direction for our patients. Dr. Gerds, I do have one quick question. Can you please explain the specific descriptions of crossover protocols?
Yeah, so crossover allows for patients to get the treatment on the other arm. So one example would be if you were on a placebo-controlled trial, one example might be the recent Momentum study with a drug called momelotinib that just finished enrolling patients. So that was a trial that was randomized between momelotinib and danazol, but it was placebo-controlled. We didn't know as an investigator which arm an individual was on. And then there was an opportunity at any point in time, if a patient's disease progressed, say their spleen got bigger by a certain margin, or after a certain period of time, things weren't better, we were allowed to unblind, not unblind the patient, but stop their blinded treatment and start on open label momelotinib. So really, there are generally two triggers for crossover. One is disease progression or lack of response at any point in time, or maybe after the initial assessment. And then usually just after the initial, they're the primary endpoint of the trial. Often they allow for crossover at that point too. So every trial is a little bit different, but those are kind of the two major triggers. So if the disease gets worse, and certainly after the primary endpoint of the trial, or often after the primary endpoint of the trial, there is an option to go to open label investigational agent. The primary endpoint is important because that is the main, really with a clinical trial, you can ask one question and answer one. You can ask a lot of questions, but you can only really answer one question. And the study is designed and powered to answer that one question. And so really you want all the patients to make it to that point, say the anemia response at week 24, or the number of people who have a shrinkage of their spleen by week 26, or whatever that primary endpoint is. You want to get all the patients to that spot so you can accurately measure the effect of the medication. So that's generally why crossover isn't allowed until after that measurement. just so we get, real accurate numbers and a real sense of how well the drug works.
I do have other questions, but I'm going to move on to our next industry partner, and then we'll come back to you, Aaron. Thank you. So now from CTI Biopharma, Sarah Buckley, who is the medical director, will share information about what's happening with at CTI Biopharma. Sarah, are you with us? Hi, Sarah. Thank you for joining us.
Let me figure out how to.
You have to share your screen. It should be down at the bottom.
Yep, I'm seeing it.
There you go. Perfect.
Okay. And I'm gonna put this into presenter view. Okay, can you still hear me and see me?
We can see it.
Okay. Hi, everybody. My name is Sarah Buckley. I'm a senior medical director at CTI Biopharma, and we're developing pacritinib for patients with myelofibrosis, and particularly for patients with cytopenic myelofibrosis, or myelofibrosis in conjunction with low blood counts, like anemia and thrombocytopenia. Just as a disclaimer, pacritinib is an investigational product. It's not currently approved by any regulatory agency. So pacritinib is a JAK2 inhibitor that also inhibits another important target called IRAK1. And not to get too into the biochemistry here, but IRAK1 is a recognized potential therapeutic target for patients with myelofibrosis who have upregulation in this IRAK1 pathway. And so pacritinib sort of is has dual targets there with JAK2 and IRAK1. Unlike other JAK2 inhibitors, pacritinib does not have any impact on JAK1 at normal therapeutic concentrations, and that may relate to some of the effects that we see in terms of being able to give pacritinib to patients with very low blood counts and achieving stabilization of blood counts. Just to give an overview of the big picture of pacritinib development, pacritinib has already been through 2 randomized phase 3 studies in patients with myelofibrosis. These are the PERSIST-1 and PERSIST-2 studies. PERSIST-1 was conducted in patients with myelofibrosis regardless of platelet count, whereas PERSIST-2 was restricted to patients with platelet counts less than 100. Subsequently, the PAC-203 dose-finding study was performed in patients who were intolerant of or who had not benefited from ruxolitinib. And we are currently enrolling the Pacifica phase 3 study, specifically in patients with severe thrombocytopenia or platelet counts less than 50. I'm going to take a little bit of time to review the PERSIST studies, PERSIST-1 and PERSIST-2, and I'll spend a bit more time on PAC-203 since that was a bit more recent, and I'll provide an update there. And finally, I'll talk about the Pacifica study that is currently enrolling. But one other thing that I should note here, and this is not pictured on this slide, but an important part of the pacritinib development pathway is that pacritinib is currently under review by the FDA. We submitted a new drug application, or NDA, earlier this year, and the FDA accepted it for priority review, and they'll be making a decision one way or another by the end of November. So we're excited, and potentially, I guess, cautiously optimistic to hear how that goes. But there's a potential for a new drug approval by the end of November. So we'll have to see. We'll have to see which way that goes. So the first the 2 phase 3 studies with pacritinib were the PERSIST-1 and PERSIST-2 study. And these studies were a little bit different. They enrolled slightly different populations. PERSIST-1 was in the first line, meaning patients did not have any prior JAK2 inhibitor exposure. But otherwise, patients of any platelet count or any hemoglobin could enroll. And patients were randomized between pacritinib and best available therapy. And at the time the PERSIST-1 was performed, best available therapy did not include ruxolitinib. And the primary endpoint there was spleen response as a marker for disease control. PERSIST-2, on the other hand, enrolled patients with platelet counts under 100. So it was selecting specifically for patients with cytopenic myelofibrosis. And patients with prior JAK inhibitors or who were currently on JAK inhibitors could enroll. So patients were enrolled regardless of whether they had had a prior JAK inhibitor exposure. And this study looked at 2 different doses of pacritinib. The total daily dose was the same. It was either 400 milligrams once a day or split in 200 milligrams twice a day. And the thought was that maybe the 200 milligrams twice a day would be more tolerable. And the control arm in this case was also best available therapy. And in this case, best available therapy did include Rux, and about half of the patients enrolling in this study received ruxolitinib as best available therapy. And here the primary endpoints were spleen reduction and symptom score benefit. The individual results of the studies can be found online, but here I'm just, since these are old studies, I'm just going to summarize the overall results, pooling both studies together. And so what we saw was that there was a benefit to pacritinib in terms of both spleen response and symptom reduction based on these 2 studies. And this benefit was seen particularly at the 200 milligram twice a day dose that was studied in PERSIST-2. So in terms of spleen response, which is what you're seeing here, there were 19% of patients responding on the pacritinib arms versus 4% with best available therapy. And for patients who received the 200 milligram twice a day dose, the response was 22%. And I'm going to be calling that dose out as I go through some of the data, because that's the dose that we are studying in our current phase 3, and that's the recommended dose. We also found that there was improvement in symptom burden with pacritinib compared to best available therapy. Over a quarter of patients met the 50% reduction in symptom score response criteria in the pacritinib arms. Specifically in the 200 milligram twice a day arm, 35% of patients met this benchmark compared to a lower percentage of patients treated with best available therapy. We subsequently did a dose finding study to determine the optimal dose of pacritinib for patients, specifically in patients with who were intolerant of ruxolitinib or who had not benefited from ruxolitinib. And this was the PAC-203 dose finding study. This study enrolled patients who had been intolerant of ruxolitinib, meaning they'd taken the drug for more than a month and they were unable to tolerate it, or they did not benefit from ruxolitinib. And patients were randomized to the 200 milligram twice a day dose of pacritinib or to lower doses to see if lower doses could be as effective. And the primary endpoint of the study was to determine the recommended dose. And this was based on an overall gestalt of both efficacy and safety. Even though we did not restrict the study in terms of platelet counts or anemia, just based on the fact that we were enrolling patients who had been intolerant of ruxolitinib or who had not benefited from ruxolitinib, we ended up enrolling a population with very cytopenic features. The median platelet count on our study was 60 with an interquartile range of 36 to 103. So even though we posed no restrictions on platelet counts, we ended up with patients who had moderate and severe thrombocytopenia. Almost half of the patients had platelet counts under 50. The majority of patients on our study also had significant anemia with hemoglobin less than 10, and quite a few were heavily transfusion dependent at study entry. Most patients had circulating blasts, and the duration of prior ruxolitinib exposure was pretty long on the study, 1.4 years. And about half of patients were both intolerant of ruxolitinib and ruxolitinib was not benefiting them, meaning people had spleen growth or failure to shrink their spleens while taking ruxolitinib. And so what we found is that the 200 milligram twice a day dose was most effective. If you look at the top graph, it's a little bit busy, but the top graph shows a waterfall plot of spleen responses with the different doses. And you can see that in the 100 milligram daily and 100 milligram twice a day groups, there were not as many spleen responses compared to the 200 milligram twice a day group, where we saw a fair number of spleen responses in this heavily pretreated population. One of the interesting things that we found is that many of the spleen responses were in patients with severe thrombocytopenia. Those patients are colored in teal rather than that navy color. And so if you look, a lot of the people whose spleen responses dip down below that 35% shrinking threshold were in teal colored lines, meaning they had platelet counts less than 50. So we saw that pacritinib, we've seen from our prior studies that pacritinib works in patients regardless of platelet counts, at least as far as spleen control. And here we saw that this was true in patients with severe thrombocytopenia as well. Among the patients who were valuable for spleen response, who had severe thrombocytopenia, at baseline, 31% achieved a spleen response. We also asked patients how they perceived their symptoms to have changed on study, and we used something called a patient global impression of change to do this, where you ask somebody, compared to before you started the drug, how do you feel on study drug or on the drug? And so we asked people after 6 months of taking the drug, how do you feel compared to when you started? And we saw that there was symptom benefit across the doses. About a third or so of patients said that they felt much improved or very much improved. But if you look at patients with describing any improvement over the course of 6 months, there was the greatest percentage of these people were treated in the 200 milligrams twice a day arm. And so this was also one of the reasons it was selected as the recommended dose. One of the interesting things we saw in this study is that the platelet counts remained relatively stable over the course of the study. We've seen similar trends on our prior studies as well, but even in patients who are heavily pretreated and who started out with very low platelet counts, we saw relatively stable trends regardless of pacritinib dose used. And similar trends were seen with hemoglobin as well. The adverse event profile for pacritinib is shown here on the PAC-203 study. The most common adverse events that we saw were thrombocytopenia and anemia, and those occur over the course of study. If you're watching patients for 6 months or a year, because you can have worsening anemia and thrombocytopenia as part of the disease itself. And so we saw this with pacritinib 200 milligrams twice a day, some of this was disease related, some of it might have been drug related, but it certainly was not the majority of patients. The data that I showed in the last slide was that platelet counts were relatively stable over time. Other than cytopenias, the most common adverse events were gastrointestinal, particularly diarrhea. This was by and large very low grade. It was manageable with over-the-counter medications, and it usually lasted for about 2 weeks and then resolved. So patients didn't need to keep taking antidiarrheal medications indefinitely. When they took pacritinib, it was really just sort of those first couple of weeks, and then things sort of settled out. So now I'd like to discuss our ongoing phase 3 study. This is Pacifica, and this is a study in patients with myelofibrosis who have severe thrombocytopenia. In this study, we're looking at the pacritinib 200 milligram twice a day dose that was determined to be the optimal dose based on our dose finding study, and we're comparing it against physician's choice therapy. So I'll go over this with you now. The main eligibility criteria for the study is platelet counts less than 50. And we recognize this area is an area of unmet medical need. A lot of studies screen out patients with lower platelet counts, particularly platelet counts under 50. And so this is an area that we see our drug as potentially providing benefit. Patients have to have a palpable spleen, since one of our endpoints is spleen reduction, and have at least some degree of symptomatology, because reduction in symptoms is an important secondary endpoint for our study. And patients may be JAK inhibitor naive or may have had a limited duration of prior JAK2 inhibitor. Randomization is 2-to-1. So 2 patients will be randomized to pacritinib for every 1 patient randomized to physician's choice. And physician's choice includes a number of common agents, including ruxolitinib. Primary endpoint is spleen reduction, and secondary endpoints include symptom reduction, the global impression of change, which is that question that asks, how do you feel compared to when you started, and survival. If you would like any more information on Pacifica, there's a website, pacificatrial.com, that I would encourage you to visit. Thank you very much.
Thank you very much, Sarah. This is great. This information is just overwhelming. And Dr. Gerds, I do have a couple of questions. Are there any trials currently recruiting for patients with MF whose platelets are around 50? I'm assuming 50.
One perfect example is the Pacifica trial, certainly
Yeah.
looking at that population. So timely question for sure.
Right.
In addition to Pacifica, there are a number of other trials that are looking at that population. There's drugs that are being developed that, like pacritinib, have the goal of not being overwhelmingly myelosuppressive, particularly within regards to platelets. So certainly we have an additional trial at open at our center looking at a different pathway altogether in a drug that's not necessarily that myelosuppressive. So there are certainly trials for that population.
I have other questions, but I'm going to move on for time's sake, and we will definitely fit those questions in. But again, Sarah, thank you very much for your time and your information. It was great. So moving along, I will move on to Sierra Oncology and to Lauren Musto. She is the Senior Director of Corporate Communications. I'm sorry I missed you in the lineup, Lauren.
Oh, no worries.
But there you are. And thank you for joining us. We really appreciate it.
No, thank you for having us on. I'm just going to pull up some slides. Okay. You can see my screen. I see a nodding head, so we'll take that as a yes.
Yes.
Thank you, everyone. Good morning, good afternoon. I really appreciate the opportunity to be able to speak with you today. And I'm excited to share a short overview of what we at Sierra are doing in the myelofibrosis space. So as Anne mentioned, my name is Lauren Musto, and I oversee corporate communications and patient advocacy at Sierra Oncology. So if you're not familiar with us, Sierra is a late-stage biopharmaceutical company, and we're on a mission to deliver targeted therapies that treat rare forms of cancer, including myelofibrosis. We're committed to taking an evidence-based approach to investigate new ways to change the cancer treatment paradigm. But we really do realize that we cannot do this alone and remain incredibly committed to the cancer community at large, because it is only together that we can transform the promise of a pipeline candidate into outcomes for patients. So our main focus right now is the development of momelotinib, which is an investigational agent for the treatment of myelofibrosis. Momelotinib is an inhibitor of 3 key signaling pathways that contribute to the major manifestations of this disease. So by inhibiting ACVR1 or ALK2, as well as JAK1 and JAK2, we believe that momelotinib has the potential to improve anemia of myelofibrosis while also improving constitutional symptoms and splenomegaly. We're currently awaiting top-line results for our phase 3 Momentum study, which we expect to have by February of 2022. The Momentum study enrolled 195 of a planned 180 myelofibrosis patients who had previously received treatment with a JAK inhibitor and were symptomatic and anemic. The trial will assess the impact of momelotinib on symptoms, splenomegaly, anemia, and the need for blood transfusions. The primary endpoint of this study is total symptom score response rate at week 24, and secondary endpoints are transfusion independence rate and spleen response rate, also at week 24. Assuming the primary endpoint is met, we will file a new drug application with the FDA in the second half of next year. Excitingly, in August, we announced that we had in-licensed a novel BRD4 BET inhibitor to expand our pipeline. So SRA515, which was formerly known as AZD5153, is a differentiated BET inhibitor through its high potency, selectivity, and bivalent binding, which may offer more complete target inhibition relative to other monovalent binders. We believe momelotinib could be the cornerstone JAK inhibitor for combination use. Unlike other approved JAK inhibitors, momelotinib is not myelosuppressive. Therefore, we believe the combination of momelotinib and SRA515 may provide improved efficacy and safety over other JAK inhibitor, BET inhibitor combinations. Our plan right now is to initiate a phase 2 trial examining that momelotinib SRA515 combination in the first half of 2022 for the treatment of myelofibrosis. And we are very much looking forward to sharing more details about that planned study as we get closer to trial initiation. So to close, I'd just like to add that we are so grateful to Anne and the entire myelofibrosis advocacy community for all the work you're doing to raise awareness and educate patients and their care partners about this disease. And we really want to especially take this moment and recognize and thank all the patients who participate in our clinical trials and all the trials that we're discussing here today. Certainly, we as an industry wouldn't be able to do what we do without your support and participation. So again, thank you again for the opportunity to share everything that we're doing at Sierra, and I hope you all have a great rest of your day.
Lauren, that was great. Thank you. It was very concise. We appreciate that. And I've followed this drug for many years. And so I'm excited about the prospect of approval in the future. I think it'll be a great possibility for our patients.
Wonderful. Thank you.
Have a good day. So we'll move on to Protagonist and to Paula O'Connor, who's the VP of Clinical Development. Paula, are you with us?
I am with you, and let's see how good I'm going to. Here we go. Share.
There you go.
And let's go to the first screen. Oops, first slide. I'm always starting at the end. All right. Well, first of all, let me introduce myself. My name is Paula O'Connor. As you've already heard, I am working at Protagonist Therapeutics, which is based in Newark, California. Unlike many of the other presentations you've heard about today, we're going to be talking about our program with rusfertide, specifically for patients with polycythemia vera. So as with many of the other therapeutics, I will start by saying that all of the information that you see today is sort of forward-looking. We are not yet approved, and as such, all that you're seeing, certainly as it relates to rusfertide, is an overview of our clinical development program. So the first slide that we have here gives you an overview of all of the programs that we have at Protagonist. We are a company that's using a peptide platform to develop new medicines specifically to treat hematologic disorders such as polycythemia vera, and then other disorders in the inflammatory space, most notably ulcerative colitis and Crohn's disease. I will not be talking about any of those today, and will be focused solely on our agent rusfertide, which is a hepcidin mimetic. Hepcidin is a hormone that is made in the body. And what it does basically is it controls the way iron is absorbed and distributed throughout the body. A hepcidin mimetic is very similar to that, but it's really designed to be an active drug. So almost like a souped up natural hormone, if you will. And in this case, we are looking at the activity of rusfertide, not only in polycythemia vera in our in a phase 2 proof of concept study, which was presented at EHA earlier this year. And then also we have another phase 2 study in which we are looking at polycythemia vera patients with significantly elevated hematocrits. That means over 48%. Rusfertide is also being developed in hematologic conditions outside of polycythemia vera, but we won't be talking about those today. On our next slide, what I have is an overview of the trial design for the study that was presented at EHA this year. It was also presented at ASH in 2020, but with a lesser extent of data. This trial was designed as a proof of concept study. So unlike many of the other studies that you've seen today, which are randomized clinical trials, this is a single arm clinical trial, and it has 3 distinct phases. The first phase, or part 1, is where we're actually dose finding for patients. So we're trying to understand what is the right dose that patients should receive to actually achieve our desired ends. Now, when we, before I go through the detail of each of these individual phases, I do want to make sure that I touch upon the eligibility requirements and then the endpoints. So this trial was limited to patients with polycythemia vera, who had a requirement of multiple phlebotomies, meaning at least 3 in the last 6 months or 6 within the last year prior to enrollment. Patients were in this first part of the study dosed with increasing doses of rusfertide based upon their hematocrit response. And that hematocrit response, meaning maintaining the hematocrit at less than 45, as well as safety, were the 2 primary objectives of this study. And in doing that, we looked very closely at a reduction in the rate of phlebotomies over time. So going back to the phases of the trial, once again, there were 3 of them. Part 1, where we looked at the dose and actually the ability of rusfertide treatment to control the hematocrit less than 45 and decrease that elevated phlebotomy rate, which all patients had to demonstrate in order to be eligible for the study. And then after part 1, we moved to part 2, which is a blinded withdrawal, where all of the patients who have been enrolled were basically randomized, if you will, to either stop rusfertide treatment and continue whatever treatment they already had when they entered the trial. And then the last phase is the open label extension. And this is where patients were reintroduced to rusfertide after this blinded withdrawal period was finished. So I think in addition to looking at the response of patients to rusfertide as it relates to their hematocrit and their phlebotomy, what sets this trial apart from many is that we also looked at how patients were feeling. So in fact, we measured the MPN SAF TSS, as well as a patient global index of change, where you basically ask patients, how do you feel now compared to previous time point? All of this data I'm not actually going to show it to you now, but certainly I would ask you to look at the EHA or 2021 website or the ASH 2020 website, and you can see the data, this preliminary data from the part 1 period of this study at those particular websites. I think the there is that in what we really saw was essentially a complete, almost a near complete reduction in the need for phlebotomy in patients who were being treated with rusfertide, irrespective of whether they had been previously treated with just phlebotomy, with phlebotomy and hydrea, or any other agents, including ruxolitinib and or interferon. So we believe that this drug has the potential to play a really important role for patients who still require additional therapy because the standard phlebotomy is not adequate to control the excessive erythrocytosis that they have with their polycythemia vera. So as I mentioned, at EHA, we gave an overview of our very preliminary results from the first phase of this trial. We anticipate that there will be additional information that will be presented later on in 2021. And I will say that on the basis of the data that we saw and presented at EHA, we have had interactions with the FDA and also with the European regulatory agencies. And the FDA has, in fact, given us breakthrough designation, which suggests that they see that this product, rusfertide, may in fact add significantly or address a significant unmet medical need for patients with polycythemia vera. In this case, for patients who are having excessive phlebotomies because they cannot have their erythrocytosis controlled with either phlebotomy alone or phlebotomy and the other approved agents, which we already know, Hydrea, interferon, and ruxolitinib. So we look forward to presenting that updated data for the phase 2 that I've already mentioned. In addition, we anticipate presenting additional data on the second phase 2 study, which I mentioned, specifically looking at patients with polycythemia vera who have very elevated hematocrits of 48 or higher. In this case, patients may be treatment naive or in some cases already on other treatments such as phlebotomy and the other agents which I've already mentioned. And then lastly, we are developing our phase 3, and we anticipate that this trial will be open for enrollment in the first quarter of 2022. So I only had 5 minutes. I wanted to make sure that I was on time, so I'm going to stop there and certainly open it up for any questions. I will once again just thank you all for your attention on behalf of Protagonist, and we look forward to partnering with you, meaning both the MPN Advocacy and Education International, as well as all of the folk who are on the line to make sure that we get this and other really important studies for patients with polycythemia vera conducted so that we know how best to treat patients. And with that, I'll stop.
Paula, first of all, thank you very much. That was great. And thank you. And it was a pleasure meeting you via, this crazy way we have to meet these days. But and I had a question for you because as I recall, was it Protagonist delivering the drug to patients who, during COVID, during the lockdown, I should say, and training the patients on how to use it and so on. I believe I'm right about that, but you can help me out here.
So Protagonist is absolutely committed to getting this study enrolled because we do see it as providing a potentially significant addition to the treatment arsenal for patients. And so we utilized just about every legal and appropriate opportunity we had to get the drug to patients who are participating on our trial. I think as many of us know, on the industry side and also on the patient side, you really do have to work together in lockstep to make sure that important questions get answered. And obviously with this pandemic, there has to be an added effort, dare I say, on both sides. And for that, I'm certainly thankful for the patients who were brave enough to continue to come into clinics as necessary to conduct the trial. And certainly we try to echo that commitment by making it possible for them to continue on the trial despite all of the challenges posed by the pandemic.
Thank you so much, Paula. I look forward to meeting you in person someday.
Yes, as do I. Anyway, thank you all for your attention.
We'll be in touch.
Okay.
Aaron, we do have some questions for you.
Sure.
Are there any trials that are specifically for triple negative?
Yeah, I think that's a great question. Most trials are agnostic of the JAK-STAT driving mutation, whether we're talking JAK2, calreticulin, or MPL. They'll include all patients. A couple are specific for certain mutations, and we may see that more in the future, particularly with calreticulin mutations, because the product of it is unique compared to the other two. But as of now, most trials, I would say, include all patients, whether triple negative or not. But there are none that I'm aware of right now that are specifically aimed at triple negative MPNs.
Okay, so wasn't there a study? I know there was actually. So I'm going to ask the question anyway, that this young woman has been to 3 different physicians with her MPN issues. And she said it's very interesting because they just look at, all 3 have just looked at her numbers and not really asked her about her symptoms.
Yeah.
And I think we did, there was a study done that said, what is the physician looking at versus the patient? And this is exactly, and it just happened to me. Believe it or not, after 17 years, my platelets were high. Never, just out of the blue. But now I know kind of why. But anyway, who did I call? Dr. Massa, and just said, what's going on? But when I was with this physician, he never touched me. He never looked at some of my symptoms. He only looked at the numbers. And so I can relate to this woman very well.
Yeah, I think, it's a tough situation because I think a lot of practicing oncologists and hematologists, may not spend that much time in the weeds of MPNs like we do. So we're at a distinct advantage.
Right.
And we know that, drugs are approved and live and die sometimes on the MPN-SAF and assessing those symptoms. And we've incorporated that into our everyday thinking. So I think, again, we are at an unfair advantage being in the weeds all the time. And I think the other disadvantage that a lot of practicing oncologists have are the fact that they have such incredibly high demands on their volumes and how quickly they see patients. Like if you got 10 minutes to see a patient, it's easy to get in the habit of just banging through things and, okay, your counselor this, we do this. And I don't think that's a justification. I think it's just more of a knock on our healthcare delivery system, not on the individual doc. I think the pressures in our medical system emphasize sometimes the wrong best for patients. I'm very fortunate to be in a system where I can spend a little more time with patients and answer those, ask those questions. I think, but I think there's also awareness things. So studies like that where there's a disconsort, we can point out the discordance between what patients are experiencing and what docs are asking about, I think are a big thing. I mean, that's not unique to MPNs. That's pretty uniform across all disease states. There was another one we did in leukemia, acute leukemia, where it was a very similar thing where we asked patients, what do you think your prognosis was? And we asked the doc, what do you think the prognosis is? They were completely different answers. And it was the same patient, same doc. They were paired paired matches, and they were obviously part of the same conversations. But I think this is not unique to MPNs either. And again, it maybe is a flaw with our healthcare system, but we know they are prevalent. We know they're important, not only for helping guide diagnosis, but also monitoring the progress over time. So basically, I think it's on to folks like myself and in your organization as well to really continue the education efforts, educating oncologists and hematologists around the world, as well as the patients, to be advocates for themselves in these instances.
Well, it's been a true test for me, I have to say. And now that patients are back into, going to physically visit physicians, I've had the great experience of having to wait till January, to actually see somebody sometimes. So, and I'm a huge advocate for myself, but anyway, okay, so I want to ask this before I forget, and that is, what are your thoughts about the COVID booster shot for our patients?
Yeah, I think that's a great question. Clearly, honestly, I think what we're trying to figure out right now, and for boosters in general, or third shots in general, is what is going on. So we know, we see that, there are some rare instances of breakthrough infections just in the general population. And is that a time dependent variable, or is it because of Delta? because both are happening at the same time. We're kind of hitting that 5-6 month mark after most people have been vaccinated, where we do see some antibody waning, but we also see the surgence of this new variant. And is it Delta, or is it a change in immune protection that's happening? And that's really the challenge that the epidemiologists are facing right now, and trying to sort those things out. And that's what they're really talking about. I think, knowing that some immune protection is waning, yes, that's where this booster idea is coming from. And we know that people who get a third shot, their immunity jumps right back up. With the immunocompromised folks and MPN patients in general, there's not a lot of very precise focused evidence, but there's certainly a lot of circumstantial evidence showing that patients with hematologic conditions in general who are on a treatment are less likely to produce antibody response. Of course, that's not the whole of the response to a vaccine. It doesn't include humoral response, but it's a part of it, right? And so, the thought is we'd rather be safe rather than sorry, because we know the consequences of a COVID infection are apparent, and they're severe, and they're long lasting in many cases. So it makes sense to err on the side of safety and boosting the protection against this virus. So I think, if you look at the current guidelines, patients who have a hematologic cancer who are on a treatment are qualified right now for a booster. And so I say, if you're in that qualifying category, it makes sense to do that, get the third shot or the booster shot. And I think for all others, it makes sense to do a booster shot as well at your 8-month mark if you don't meet that narrow definition. So with every patient who bring, we see in clinic, we're kind of bringing up this construct where the definition is pretty loose from the CDC. It gives a lot of leeway, but then that offers an opportunity for people to get protected when they need it.
Thank you. Okay, so we'll move on to our next industry partners presentation. This is Jiran and Soria Doherty. Are you with us? There you are. Hello.
Hi, how are you?
Good, thank you.
Let me go ahead and try and share my. Thank you for having me. I know that Timra was supposed to be here today, and she's our study physician. And then I am now joining to speak with everyone. But before I get started, I'd like to say thank you for all the great work the MPN Advocacy Group does to bring patients and caregivers and physicians and pharmaceutical companies together. So this is a great opportunity. So I'm Soria Doherty. I'm the Executive Director of Clinical Development here at Geron. And as I mentioned, our study physician is Timra Berry. And we wanted to take a minute to let you know about our ongoing phase 3 study. But before we do that, we can talk a little bit about Geron. And Geron is a late stage pharmaceutical company developing imetelstat, which is an inhibitor of telomerase, which is an enzyme that is overactive in blood cancers and for which we are treating hematologic myeloid malignancies. We did complete a phase 2 study of imetelstat in intermediate 2 and high-risk myelofibrosis patients who were relapsed after or refractory to JAK inhibitor treatment. And in this study, I won't go into detail, but you are very welcome to look up the ASH data that we have recently presented and the JCO data. But in this, we showed a potential for survival benefit and an encouraging symptom response rate, which brings us to our ongoing phase 3 study, which I'll show you here. So we recently opened this study. It's a phase 3 study in intermediate 2 and high-risk myelofibrosis patients who are refractory to JAK inhibitor treatment. And this study is known as the MYF 3001 study or the Impact MF study. And it's an example, or it's an open label randomized controlled trial comparing imetelstat to best available therapy. So it's any therapy that you can receive other than a JAK inhibitor therapy, since we're enrolling refractory patients. And you would be randomized to one of these arms, 2-to-1 randomization scheme, and treated. So the primary endpoint for the study is overall survival, and symptom response rate is a key secondary endpoint. And we plan to engage in over 100 sites to open globally. And we can find details about the study on the geron.com website, as well as clinicaltrials.gov. And we look forward to working together to improve the lives of MF patients through this trial. And that's what I have. Thank you.
That was quick, short and sweet. Thank you. Well, thank you. And, you know, I hope to meet you in person also someday.
Great. Thank you. Same to you.
Thank you. Okay, Aaron, so we have some more questions for you. This has turned out really well because
Fantastic.
we were able to ask you these questions. Let's see. With the high incidence of symptoms such as bone and muscle pain, gout, difficulty sleeping, this is a question we get asked a lot. Will some of these new drugs alleviate some of the constitutional symptoms?
Yeah, I mean, really, that's the goal, right? For a lot of these treatments. So if you think about some trials, their primary endpoint, as discussed today, really are measuring spleens. And that's somewhat practical. It's pretty easy to do a CAT scan or an MRI or an ultrasound and measure a length or a volume and then see if it got bigger or smaller. I mean, it's very precise, it's easy to measure. And that's why a lot of these clinical trials have that as a primary endpoint. But as a key secondary endpoint, or even as a co-primary endpoint, symptom burden is often included. Symptoms are a little bit more difficult to measure. They are subjective. No matter how you slice the lemon, they're subjective. And a lot of times when we try to analyze the data, you see that some people are getting better, but the numbers are kind of muddled or gobbledygook. You know, like, for example, like if you're feeling better, like your night sweats improve on the medication, but then you get an infection, yeah, your night sweats might get worse because it's from the infection, not from the, but that you still report it as night sweats, but that muddles your results. So because it's multifactorial and subjective. You know, one of the trials that we discussed today mentioned this patient global impression of change, which I think is a nice way of kind of cutting through a lot of that. Are you better or worse or the same since starting this medication? It kind of cuts through the mess, which is really nice way of looking at things. So, but at the end of the day, if we're deeply affecting disease, if we're suppressing the cytokines, if we're even killing off the clones or reversing the fibrosis, we would also expect, as a reflection of that, improvement in symptom burden. So just because it's not the primary endpoint of the trial, it's still a goal of the therapy. It's just because the messiness of measuring it makes it difficult to be a primary endpoint.
So I know, I don't know any MPN doc that has said for patients not to get the flu shot, but I was injected in February, so my 8-month mark is coming right up. But for those patients who also need, or should get the booster shot, who typically get the flu shot in October and November, what would you recommend to them?
Certainly, I'd recommend the flu shot. In a typical year that, we're not wearing masks and social distancing, the flu can kill 20 to 40,000 people per year in this country. And the flu shot can protect against the more severe strains. There's also emerging evidence that there is a concern that this year's flu season may be much worse than last year. The flu was non-existent because we were all social distancing, but there's worry that it might be worse this year. Or they may see a lot of co-infections between COVID and flu, which are obviously really bad. We're seeing that in kids with co-infections of COVID-19 and RSV. So certainly I would recommend getting the flu shot. It's like voting in Chicago. Do it early and often. Well, at least early. What my current guidance though is to kind of space them out I would give yourself at least a week between them. I wouldn't wait months between them, but give yourself a week, because if you do have some kind of low grade temps and achiness from the COVID vaccine, and then you're also going to have that from the flu vaccine, you're going to probably feel doubly poorly during that immediate post-vaccination period if you do them at the same time. So spreading them out just makes it not so onerous. But I would not delay by any significant margin of time.
Okay, that's good to know. Very good to know. Okay, well, we'll hear from our next partner, Constellation, and Sean Yi, sorry, who is the field medical director for Constellation. Hello, good morning.
Hi, good morning.
How are you?
I am good. I just want to make sure I have the right background. I'm going to share my screen.
Okay.
Let's see. Pardon me? Can you see?
Yes, very good.
Perfect. So thank you again, Anne. Again, I am Shangyi Liao, Field Medical Director at Constellation Pharmaceutical Medical Affairs. I just want to give you a brief update on our trials today and also background on our company. So we are a Massachusetts-based company with over 12 years of discovery and development history, focused on the epigenetics and cancer biology. Our drug candidates, which were all internally discovered, are the product of our expertise in gene targeting for diseases where gene regulation and control matters. As of July 15, 2021, Constellation Pharmaceutical is now a MorphoSys company, and we are committed to delivering breakthrough therapies that bring hope to patients. The lead compound that generates Constellation is an investigational BET inhibitor, pelabresib, also goes by the name of CPI-0610, that we are developing as potential treatment of myelofibrosis. Pelabresib is a novel orally administered small molecule designed to selectively inhibit the function of BET protein to decrease the expression of abnormally expressed genes in cancer. Please note that pelabresib is an investigational new drug and is not yet approved by any regulatory agency. Constellation is currently conducting a phase 2 clinical trial of pelabresib in myelofibrosis and essential thrombocythemia, and a larger phase 3 study called MANIFEST-2 has initiated for myelofibrosis. MANIFEST-2 is a randomized, double-blind phase 3 study in adults with myelofibrosis who have not been previously treated with a JAK inhibitor. The purpose of the study is to evaluate the safety and the efficacy of pelabresib plus ruxolitinib, a JAK inhibitor, compared with ruxolitinib alone. For more information, you can find both the MANIFEST and the MANIFEST-2 trial at clinicaltrial.gov, or check out our website at www.constellationpharma.com. We all recognize that we need new therapies for myelofibrosis patients that addresses the areas of unmet need, and we are working hard to develop such a therapy. At Constellation, our clinical and research team are deeply invested in a scientific approach to bring therapies to patients. Our interest and commitment to patients and caregivers extends across the MPN community, as we have additional plans for pelabresib and early stage programs that may have utility in myelofibrosis and other MPNs in the future. I applaud this community of patients and caregivers suffering from the consequences of MPNs. It truly inspires us to work every day to develop these new therapies. And a final thank you to Anne, your group, Dr. Gerds, and most importantly, the patients. Thank you.
Another very short and concise presentation, but we're really looking forward to these new drugs and the progression of the drug and where it goes, and hopefully remains in the MPN community until commercialization, because we need them desperately. So thank you very much. It was good to see you. We'll move on to Pharmacentia and Dr. Ray Urbanski, who is the head of clinical development and medical affairs there. Are you with us, Ray?
I'm here, Ann. How are you?
Hi, Ray. Good to see you.
Long time no see.
Yeah, yes. Thank you for joining us today.
Oh, it's my pleasure. So let me share my screen. Can you see that?
We can.
Okay, so I am going to basically go over some of our studies. One in particular was the study that we're doing with ROPEG interferon alpha 2b and essential thrombocythemia. But before I do that, let me just show you a little bit about what ROPEG actually is and what it looks like. So ROPEG is a monopegylated interferon alpha 2b, which differentiates it from some of the other second or first generation interferons that are currently out there, which are polypegylated. The monopegylation leads to rather distinct and beneficial PK/PD sort of characteristics that allow patients to dose every 2 weeks and sometimes every 4 weeks. The original program for ROPEG interferon was in polycythemia vera, and here's just a quick overview of 2 of the studies that were done. One was a PEG and vera, which was a dose finding study, and the other one was a pre-planned single arm study which we called the PROUD PV, and then the CONTI PV, which is actually short for the continuation PV, which was a rather long phase 3 study. Of note is that the very long follow-up time with these patients in the PEG and VERA study was over 5 years. In the continuation PV was well as over 5 years. And all of this data has been published and presented at either ASH or in some top tier journals. Just as quickly, just on some of the data that we saw with the PEG and with the PROUD CONTI study is when we looked at 12, 24, and 36 months, we saw around the 70% CHR rate, which is fairly significant. Overall, when you looked at the objective response rate or the overall response rate, it was around 84%. This was consistent with the PEG and VERA study as well. So again, quite indicative of sort of deep and durable CHR responses. More importantly, when we looked at allelic burden or molecular responses, basically trying to assess what the drug was doing at the level of the bone marrow to actually affect the JAK2 mutated cells versus the total, we saw significant effects with molecular responses over time. And you can see that here with the 68, 66% at 24 and 36 months. How we define molecular response is shown on the left-hand side of this graph, which I won't read. But again, a clear indication that this drug was working at the site of the bone marrow and actually reducing JAK2 mutated cells. So from there, we went on to take a look at what would the drug do in essential thrombocythemia. So we explored these treatment options, looking at the outcomes, basically hematologic responses. We were interested in the safety profile. Again, some of the clinical data from the early study showed that it had a better toxicity and tolerability profile, less frequent dosing, and basically looking at if we had disease modification of preventing some of these diseases from going to MF or AML. The study goal for the SURPASS-ET trial, as we call it, was to evaluate the safety and tolerability, as I mentioned, as a second-line therapy for ET. This is basically second-line to hydroxyurea resistant or intolerant patients. It's an open-label randomized design. I'll show it to you shortly. Study duration's about 14 months. You show there the screening, 52 weeks of treatment with a 4-week follow-up, which is sort of standard. The patient population is those who have tried hydroxyurea and it's no longer effective, or they actually have side effects. This slide's actually a little bit dated. It says we have 50 centers in the US, Japan, Taiwan. You can read the rest of the countries. We're actually opening up sites in Canada and Europe as well. The sample size is dated about the same, around 160 participants to get 130 evaluable. This is the study schematic. Again, eligible high-risk ET patients, resistant or intolerant of HU, interferon naive, or actually, if they were treated with previous interferons, had to be interferon antibody negative, and a platelet count of greater than 450. We stratified the patients based on country, TSS, and platelet count. Obviously, with the addition of Canada and Europe, the stratification by country is going to be modified slightly. You can see it's a parallel group study looking at 250, 350, 500 dose titration of ropeig interferon, which is a little bit different than it was in the previous trials. And it was going to be compared to anagrelide and basically utilizing the labeling, the labeled doses. And I already showed you the duration of the trial on the previous slide. So the main inclusion criteria is here. And again, when you're looking at the high risk, it's either older than 60 years and JAK2 positive, or they actually had a previous thromboembolic event. I already read the rest. One of the things of note is the platelet count had to be greater than 450, but also we're looking for patients with an elevated white count of greater than 10. This is one of the major factors leading to sort of recruitment being less than ideal. But we're working extensively with our investigators to see how we can sort of improve that. Main exclusion criteria are listed there to the right. The primary endpoints you can see here, primary and secondary. Primary is response to treatment as defined by the modified ELN criteria, and that's listed on the left. And we're also looking at the MPNS TSS, which basically multiple presenters before me actually went over some of these measurements. Secondary endpoints, we're looking at durability of response as well as the occurrence of thromboembolic events, et cetera. Hold on one second. So we encourage and appreciate the spread about and the word about the trial. We are listed on the clinicaltrial.gov site. We also have our own microsite, which you'll see contact information for those of us at Pharma Essentia, as well as some investigators. We also are passing around and providing study information for both patients and HCPs. And this is also available on our website if you want to download it. So with that, Ann, I know that was brief, but hopefully I covered the requisite information that you're looking for. So thank you.
You did, and I do have a question for you about the difference between ROPEG and the pegylated interferon Pegasus, if you can share that.
Yeah, certainly. So, drugs like Pegasus, Peg-Intron are polypegylated, right? So they have anywhere from 8 to 14 different pegylated isomers. This seems to alter the pharmacokinetics somewhat differently. And so you get a different PK/PD, PD, and what initial studies seem to show is a different tolerability profile. Now, as you know, and we have not done a head-to-head with ROPEG versus Pegasus, so I can't give you any head-to-head data. So I can only just tell you what we've seen in our studies. And again, the only thing I could say definitively is that when you look at the structure and the number of pegylated isomers in the PK profile, that seems to have a difference in the exposure of the drug to the patients and its effect on the drug, as well as its tolerability profile. Hopefully I answered your question.
Pretty much. Thank you so much, Ray. We really appreciate it. Good to see you again.
Same here.
Stay safe. And Dr. Gerds, when should a patient consider interferon, and who is that patient? Can you give us kind of the description, or what's going on with that patient that you would suggest interferon?
Yeah, so I think interferons, carve them out in the MPN world in patients who don't have large amount of myelofibrosis. So they've been looked at as a class of drugs in patients with, like, say, grade 2 or 3 myelofibrosis. And certainly that population seems to have less of a benefit, when looking at interferons as a single agent. So generally we're thinking about PV, ET, and then maybe prefibrotic myelofibrosis is kind of the range there. In PV and ET that we do, here in the United States, we have pegylated interferon that's commercially available as of right now. And then we have one kind of prospective randomized trial, the MPNRC 112 study, that looked at that in the upfront setting, and the 111 study that looked at it in the second line setting. And that included both PV and ET patients. And at least in the upfront randomized trial, it was on par with hydroxyurea when we both look at efficacy and side effects. So it makes sense to have that as, like, kind of choice, hydroxyurea and interferons as choice 1 and 1A, really, based on that trial. And if you look at guidelines like the NCCN guidelines, it really kind of outlines that. For people who've already had hydroxyurea, it is a great second line therapy, again, as demonstrated in the MPNRC 111 study. And then, someday we hope to have ROPEG interferon here in the United States, as mentioned, and that would provide another opportunity, depending on what the label looks like, to get interferons to patients. There is a subset of patients who have prefibrotic myelofibrosis. They don't have the high grades of scar tissue in the bone marrow, but definitely have myelofibrosis, and we can sometimes use interferons to improve symptoms. Lastly, again, there is the notion that interferons can lead to deeper remissions in some patients, particularly molecular remissions. And I think that is an attractive option, particularly for younger patients. So I think, younger patients, with that in mind, also with the reproductive considerations of hydroxyurea, often think about interferon as a great alternative to treat their ET or PV or prefibrotic myelofibrosis.
Okay, and so, we're kind of wrapping up now, a little early, because we had, a few of our partners really did a great job of editing, I think, and just, very concise presentations. And of course, some of their drugs are not at a place where they can expound on what's happening. in detail, but very good presentations by all of them. Thank you so very much, and for their continued support so we're able to do these programs. We will be hosting another one tomorrow for MPN physicians, and these are physicians that are not only MPN specialists if they want to join us, but these are physicians that see MPN patients that may be in remote parts of the country and the world, actually, that maybe have one or two patients and don't really know what's going on with clinical trials, and would be very interested in knowing. So that will be tomorrow, the same time, and Dr. Ron Paul from Memorial Sloan Kettering will be playing the role of Dr. Gerds tomorrow. And so Dr. Gerds, is there anything you would like to say as we wrap this up?
Yeah, I mean, I think, today was just a small menu of all the things going on in the MPN world in terms of clinical research. Clinical research is the tool that we use to take this discoveries that happen in the lab about these diseases and move them forward into the clinics. And so I think it's incredibly important, not to be a downer or anything, but as of right now, there are very few drugs that are commercially available for the treatment of MPNs, that have regulatory approval, or even fewer. So, this is not like multiple myeloma. I use an example because they have like 1 million drugs. They have a new drug like every day. It's ridiculous, but it's fantastic for multiple myeloma patients. But that's the point we're trying to get to.
Right.
There was the not to draw upon the MPNRC again, but there was the adage that someone mentioned at a meeting that the goal of the MPNRC is to basically treat and or cure patients without having to do a transplant. Not that transplant isn't a fantastic option for many patients, but really our goal is to push the field forward even more. And that's and the tool we do that through is clinical trials. So I certainly encourage folks to search the interwebs. Clinicaltrials.gov is one way of doing it. Individual hospital websites often have their clinical trials posted. Websites and groups like MPN Advocacy, as well as the MPN Research Foundation, have their trials listed. I have trials that are open listed as well. So certainly, in addition to researching the disease and the disease process and what may be going on, I think it's worth considering clinical trials and talking to your doctor about them.
I agree, and I thank the people, the individuals that do get into the clinical trials. God love them. I mean, it's quite a big deal, and
without them, we couldn't do better.
That's right. That's right. We wouldn't have anything on our shelves for any illness. So I thank all of our partners, all of the people that joined us this morning. I thank you for your support as well. And Dr. Gerds, as always, we miss you. We miss seeing you in person, and we really appreciate your time and input. It's always very much needed, and thank you so much.
My pleasure.
Take care.
Thank you.
Be safe, everyone.
Thank you, Ann. Thank you, Dr. Gerds.
Bye.
Bye.